So immuno-oncology is really about modulating the immune response and using the immune response to combat the tumors. CGI can support clinical trials by bringing to the table expertise that we have in-house and also offering a variety of platforms to select from, including morphologic assessment, flow cytometry, immunohistochemistry, cytogenetics, and molecular, including next-generation sequencing. There is a lot of expertise, particularly in the field of hematologic malignancies, but also solid tumors, that allows us to engage early with pharmaceutical companies in designing the clinical trials, but also in developing the tests that can assist and help understand the mechanism of actions of the novel drugs. In order to understand the effect of the new drugs on the tumor, it is important to assess not only the effect of the drug on the tumor, but also on the immune cells or the tumor-infiltrating lymphocytes. And in order to do that, often it is very helpful to look at the in situ at the tissue. So this is where our expertise in histology and morphologic assessment comes into play. We have multiple new antibodies that we developed and optimized for the use in immuno-oncology clinical trials. One of the examples of such antibodies is a very important biomarker that came to the forefront of attention. It is PD-1 and PD-L1. We have various tests that we've developed and use in different malignancies in order to assess the expression of PD-L1 on the tumor surface, but also on the tumor-infiltrating macrophages and tumor-infiltrating lymphocytes. In addition, we have various platforms. We are platform agnostic, so we can use either DARCO or Ventana platform to assess expression of that new exciting biomarker. We developed a new immuno-oncology panel that utilizes a 10-color flow cytometry, and because it is possible to look at up to 10 markers on one specific population, it's possible due to that to detect the rare populations of cells. So in the context of immuno-oncology clinical trials, we had multiple requests to be able to detect populations that appear at the sub 1% frequency in the peripheral blood or in the bone marrow. So our panel allows looking at natural killer cells, macrophages, a specific specialized population of T cells like T helper and T regulatory cells, memory and effector T cells. The next-generation sequencing platforms allows us to look at the genomic events and also events that take place at the gene expression level. So at CGI, we have a variety of platforms that allow genomic and gene expression profiling, including Illumina, Ion Torrent, and microarray-based, Affymetrix-based platforms. It is important in the context of the immuno-oncology drugs because it allows us to accomplish several things. First, to profile the certain genomic events like mutations or translocations that may affect response to the drugs, but also what became evident and clear based on the published literature and the recent studies is that tumors evolve a lot. And because tumors evolve due to the mutations, there are certain neoantigens that tumors present. And this is how tumors are able to evade the immune surveillance and evade eradication by our innate immune system. The assessment of the neoantigens is a new area, and next-generation sequencing technology allows sequencing and profiling large number of genes and defining how tumors evade the immune surveillance. We can also contribute to clinical trials by using our cytogenetics lab, which offers conventional cytogenetics and also a large portfolio of FISH probes. At CGI, we are excited to be part of the immuno-oncology trials, contribute our expertise, and improve treatment for cancer patients.